Hi guys. So today we are going to discuss special senses. And for us to discuss special senses, we need to kind of figure out where special senses come into play in our body. And so for us to talk about it, we need to start a little bit with the nervous system. The nervous system is the body's control center and communication network. And so how the nervous system functions essentially, the nervous system basically monitors changes, and those changes are what are known as stimuli. They can occur inside or outside the body, and that stimuli is known as sensory input. And so if you look at this picture right here, you notice that you have your eye, and basically this eye is taking in all the information around it, and that is just this first part of it, right? It's just taking it in. Now, what our body basically decides to do once it takes it in is the function of the nervous system. So as you see in part 2, it says processes the sensory input and makes decisions about what should be done. So your eye is going to take in the surrounding, and then it's just going to communicate that information to the nervous system. And basically the nervous system processes what those sensory receptors are telling it, and then it does what's known as integration. And this is basically, it makes a decision. And then it initiates a response. So this is, in this example, motor output, right? And it initiates response by either activating muscles or glands. These are effectors. And so if there is a ball coming towards you, the sensory receptors are picking up on that through the process of changes with photoreceptors. And then that information gets sent to the brain. And then the brain is going to figure out, okay, what do we want to do with this? And if you're playing kickball, you'll want to kick that ball. And so then essentially what it's going to do is it's going to talk to the effector muscles in the leg for you to kick the ball. Now, the special senses basically use special sensory receptors to bring in information from the inside or outside of the body. These are distinct localized receptors, and they are held in the head. And so the special senses that this chapter talks about is vision, taste, smell, hearing, and equilibrium. Now, if you look, 70% of the body's sensory receptors are in the eye, and that is just a part of vision. Right? So a big chunk of them are involved in what we see. You have taste and smell, and then you have hearing and equilibrium. And equilibrium involves the ear as well as hearing. Just to give you an idea, about half of the cerebral cortex in the brain is involved in just visual processing, if that just gives you a scope of how important vision is. And so for this particular lecture, we're going to focus on vision, hearing, and equilibrium. It's kind of what we focus on in lab. And so the muscle of the eyeballs are very, very important. They have a lot of functions to them. They basically enable the eye to follow moving objects. So when you're looking around, making sure that balls are being thrown, if you play softball or a fly is bothering you around your head, basically your eyes are able to move around due to these basically 6 strap-like extrinsic eye muscles, right? They also help to maintain the shape of the eyeball, and they hold it in orbit. And so these are the 6 muscles. Note that you don't need to know the controlling cranial nerves here. For this one, we will talk about the different cranial nerves and how they're important, and you will need to know them. But just as of right now, it's not important that you know specifically what muscle is controlled by what cranial nerve. You will need to be able to label the muscles and know their muscle actions. So let me actually go to the next picture so that we can look at the eye a little bit better and talk about their movement. So here are the extrinsic eye muscles, and let's start with the lateral rectus muscle. And so rectus, you'll notice that it's attached up here in the front, right? And you can see that the fibers here in the muscle move this way. So anything that's attached this way and goes, so if you look at this guy too, that's rectus. This one that's attached down here is rectus. And then you have one on the other opposite side of the eye. They're all rectus. Now with this, so remember how we learned about muscle. Eye muscles are no different than regular skeletal muscles. And so remember, muscles don't push, they pull. So if you look, we're attached over here. So what happens if this muscle pulls and gets smaller? That will tell you how it moves the eye. You see it's on the lateral side. This is our nose here, right? So this is towards the outside, or the lateral part of our eye, which is why it's called lateral, right? And basically, if this guy pulls this way, right, it's going to move the eye laterally. Let's look at the other one that's on top. So here, right, you notice it's the top and it's moving this way. So you have the rectus, and at the top is superior, right? Now think, if you have this guy and it pulls this way, which way is the eye going to move? It's going to move the eye, right? It's going to elevate the eye. Now, because of its location as well, it can, on top of the elevating the eye, because it's a little bit closer here on the inside, this superior rectus is also going to make it go medially. Now, if you go towards the bottom, this guy again here is going back, so your rectus, and it's on the bottom, so it's inferior. And so if this guy pulls, right, it's going to move the eye, or it's going to help move it so that it depresses the eye. On top of that, because of the way that it is moved over here, it's going to also make it go medially. And what's great is when we're in the lab, you'll get the chance to actually see these. Now, you do have one that's on the inside here. And that inside one, again, it's rectus because it goes straight back. And what do you think it's called? It's on the inside towards the midline, so it's the medial rectus muscle. And that, because it's on the inside, when it shortens, it's going to make the eye go medially. Now, please don't write left, right on these, right? Because depending on which eye you're looking at, left and right is going to be different. So make sure you're always using these terms. Now, if you notice here, these guys are going sideways. And the sideways is oblique. So the inferior oblique is going to, it pulls it from over here and makes it shorter. So because it's attached towards the back, it's going to be different, right? Because all the other ones have been attached in the front. So if you pull at the front, they're going to go in the direction that they're pulling. because it's attached in the back, it's opposite. And I'm going to show you this in the lab just so that you can get a better idea. And I have a video that I'm going to show here. So for the inferior obliques, it's going to elevate the eye, which is up, right? And then it turns it lateral. For the superior oblique, basically the superior oblique is going to depress the eye and then again turn it lateral because it's connected on the back.
Hi, understanding the actions of the extraocular muscles can be frustrating and confusing, so I hope this video helps. You may also want to watch a video on eye movement terminology before this one. The eye sits within the bony orbit. The orbit has a roof, a floor, a medial wall and a lateral wall. It is therefore somewhat pyramid-like in shape. The two orbits have medial walls that run parallel to one another. The lateral walls are at about 45 degrees to the medial wall, or 90 degrees to one another. The eye sits within the middle of this space, with the four recti muscles converging on the orbital apex, where they insert into the annulus of Zinn, a fibrous ring of tissue that encircles the optic nerve. These recti are superior rectus, medial rectus, and inferior rectus, all supplied by the oculomotor or third cranial nerve, and lateral rectus, supplied by the abducens or sixth cranial nerve. The inferior oblique is also supplied by the oculomotor nerve, while the superior oblique is supplied by the trochlear or fourth cranial nerve. Contraction of the lateral rectus abducts the eye, while contraction of the medial rectus adducts the eye. The two opposing muscles are described as being antagonists. Sherrington's law of reciprocal innovation states that when a muscle is stimulated, its antagonist is inhibited. This means that abduction is a combination of contraction of the lateral rectus and relaxation of the medial rectus. As well as having an antagonist muscle, there is also a yoke muscle which achieves the same movement in the other eye. So for right gaze or dextroversion, the lateral rectus abducts the right eye, while the medial rectus adducts the left eye. Herring's law states that yoke muscles receive equal and simultaneous innervation, the magnitude of which is determined by the fixing eye. When the eye is abducted 23 degrees, the superior and inferior recti are aligned with the axis of the eye. They therefore act purely to elevate and depress the abducted eye. The superior oblique acts to intort, and the inferior oblique acts to extort the abducted eye. When the eye is adducted, the inferior oblique and superior oblique become the main elevators and depressors. The superior rectus now primarily intorts, and inferior rectus primarily extorts. How can you remember these eye movements? Well, I draw a simple diagram. This diagram shows the abducted eye due to lateral rectus, with elevation from superior rectus and depression from inferior rectus. It also shows adduction from medial rectus, with elevation from inferior oblique and depression from superior oblique. Finally, for the intorsion and extorsion, simply turn the eyebrows into arrows. The superior rectus and superior oblique muscle are both intorters, which act in the direction of the arrow you have drawn.
So hopefully that helps you with the movement of the eye. Now the eye must stay lubricated. And so we have what's called the lacrimal apparatus, and that includes the lacrimal gland and ducts that drain into the nasal cavity. And so the lacrimal gland is located right above the lateral end of the eye, and basically its function the function of the lacrimal gland is basically to provide tears for the purpose of protection. And lubrication. And basically that helps to keep the eyelid from sticking to the eyeball. And so essentially what it does is these tears, also known as lacrimal secretions, basically are dilute saline solutions. They contain mucus, antibodies, and lysozyme. And so essentially, those are all things that help to protect the eye from getting things like infection. And making sure that all your cells there also have the appropriate ions to function, and that if there's anything there, that it can break it down, right, or get rid of it. So basically it gets secreted from the lateral part of the eye, and so when you blink, the blinking is actually spreading the tears towards the medial commissure, which is towards the middle of the eye. And so what we're going to talk about, we're going to look at the pathway that it follows tears whenever it leaves that lacrimal gland. So let's go to the next. So if you look here, right, you can see that this is the lacrimal gland. And then coming out of the bottom of them are the ducts here. So, essentially what happens is that the lacrimal gland again produces tears, and it's going to go through these excretory ducts of the lacrimal gland to go into the eye here. And then essentially it gets moved across the eye towards the medial area, and that happens during blinking. And then we have this area that's right here, the lacrimal punctum. This is where the tears exit. So it goes in here through the lacrimal caniculus down until we get into the nasolacrimal duct, and then it continues into the inferior meatus of the nasal cavity. So all the time you're producing tears, and basically the tear production, put this maybe a different color. Tear production equals the flow or the exit through the punctum. So that's why you don't have problems with your eyes running or tears coming out. Now what happens whenever we get emotional or whenever we have items that get stuck in our eye, we end up producing a lot of tears. And so much so that it starts to basically run out of the eye. Now we still get some that go down through that area, which is the reason why whenever you cry, your nose runs. Because you have way more tears or this lacrimal secretion going into your nasal cavity than you normally do. And so that essentially kind of explains tear and tear production. Now, there are homeostatic imbalances for this, right? Imbalance. And an imbalance for this is what we know as chronic dry eye. The cause is unknown. It's associated with computer use, LASIK eye surgery, postmenopausal women. We didn't have enough to worry about women, and then this can also come from being postmenopausal or the use of allergy meds. Things that have antihistamine in them. And so chronic dry eye just means that there's a result in improperly made tears or not enough tears. And when you don't have tears, you you don't have the secretion there, and the secretion has things like lysozyme and antibodies. So you become at risk of complications or like things like eye infection or scarring of the eye because things start sticking together. This can essentially cause problem with vision. Now let's talk about the structure of the eyeball. We've talked about the type, how we move them and how we protect it. So now we're going to essentially go over the structure of the eyeball. Now, I review the structures here, but I'm going to actually point them out and talk about them on the picture itself. But we're going to talk about the wall of the eyeball and the eyelid. That's the conjunctiva. The wall of the eyeball actually contains 3 layers. It's the fibrous, the vascular, and the inner. We're going to go into and subdivide those. The cavities are filled with this thing called humor. And basically, we're going to look at that when we dissect an eye in this class. You're going to notice that the lens is going to help separate the cavity. So we're going to have an anterior and posterior part of the eye, anterior being in front of the lens, posterior being behind. And then we're going to talk about the retina. And this is the internal layer of the posterior cavity, and this is going to be made up of a couple layers, and it's going to have a lot of 250 million photoreceptors in this thing called basically a blind spot. So let's go through and talk about the different layers here first. Let me see this really quick. And so let's start. You don't really see it on here, but we are going to start here. So we have the first thing, which is called the conjunctiva. And basically this is just a thin, clear mucous membrane. And this is the covering. So this is the outermost covering of the eyelid. And the outer white surface of the eye. So when we're talking about this, it's going to be towards the very outside, and it helps to provide lubrication. So that's different. We're about to get into these different areas right here. So that would be on the very, very, very outside of the eye. So now let's get to this outside fibrous tunic, which is the fibrous layer of the eye. It's the outermost layer, okay, and it's made up of dense avascular, which means it doesn't have a blood source, right? Connective tissue. I'm going to put this on the top here. So it has 2 regions. If you can see this, it has the sclera and it has the cornea. So the sclera is the posterior portion of the eye, that's the covering. And the sclera is important because it helps to protect, shapes the eyeball, and it anchors the extrinsic muscles. Right? So basically, from that part out. And so the cornea is this guy right here. The sclera is posterior and it's opaque. where the cornea is transparent. You can see through it, right? And it's anterior, of course. You're hoping it's anterior because this is what you're seeing. This is what's allowing light to go through. It's going to bend light as it enters. This area also has pain receptors in it, so if something happens, it gets damaged in some way, that's going to contribute to the blinking and the production of tears. So that is the fibrous tunic or tunic. The next part is what's known as the vascular layer. And it's made up of 3 parts: the choroid, the ciliary bodies, and the iris. And as you can see, this choroid here is right there. And this is the posterior portion. This guy is vascular. So on here, when we look at it, this is the area that provides blood. Now this guy is also, put it right out here. It also has a brown pigment that's part of it that helps to absorb light to prevent light scattering, and what's known as visual confusion because you have too much light there. So that's a choroid. The ciliary bodies are right over here. See them here, and you can see them there. And these are rings of tissue surrounding lens. So this guy is on the anterior part, as you can see. And so what is this? They're basically smooth muscle bundles. And you also have ligaments there. And this is going to hold the lens in place. And it's going to control lens shape. And that should be apparent because you can see all these little things connecting to the lens. And then the last part of this is the iris. And so this again is in the anterior, right? And we know it as the colored part of the eye. Right? So it's normally around here. Now, in this middle, that part that's right here, that's what's known as the pupil. That's a central opening, and its function is to regulate the amount of light that will enter the eye. And they do that by basically having 2 types of muscles. So let me actually do this really quick. so the pupil, when you're looking at close vision or bright light, you're looking at the sphincter papillae that is regulating it. And this is a circular muscle. So this contracts. Basically, the pupils will constrict. When we're talking about distant vision or dim light, we're looking at the dilator pupilla. And this is a radial muscle. So different shape, right? And so when this guy contracts, then you're going to have the pupil dilate. So that's how basically our pupil is controlled. Now, your pupil can change size. Specifically, it can dilate during emotional states. So it's not just the amount of light coming in. So if you find something you're looking at appealing, or if you're requiring problem solving, both of these can cause your eye to essentially dilate. And so let's look at this last part here. Here you'll notice we have the retina. And the retina has both pigmented layers and neural layers there. So before we get to this, let me just finish this last part talking about, because we talked here about the pupil, but we never talked about this. What is that? That is the lens, right? And so this is biconcave. What did I say? I sat there and did that. Biconvex, not cave. It would go inside if it was biconcave. Sorry. So it's biconvex. It's transparent, which means you can see through it. It's flexible. When we're seeing, we're changing the shape. It's known as accommodation, so that you can manipulate the light to have it focus differently in the back of your eye so that you can see better. And it's avascular, which means it doesn't have any blood going to it. So it does. It changes shape to allow light to go through it and focus here in the back of the eye, otherwise known as focusing light on our retina. So you can have a homeostatic balance of this guy. It's known as a cataract. And we're going to talk a little bit more about cataracts when we get to that part of the lecture. Now, here we can't see it, but inside here and right here, there's this jelly-like substance, and it's called humor. Now out here, it's called aqueous humor, and think A for anterior. So in this, aqueous humor sounds like aqua or water. 98% of it is water, right? And the rest are enzymes, electrolytes, and sugar. They're continuously produced by these things called capillary bodies. And it's important because that's what's nourishing the part of the eye that doesn't have any blood that goes to it, otherwise known as avascular, and it helps maintain the pressure. Now back here is what's known as vitreous humor. So this is in the posterior. It's gelatinous consistency, so it's a little bit more like gel than what you get in the front, and this helps to maintain the shape of the eye. And basically, it causes the compression of the retina and it helps to basically protect the retina. It's only produced once. And it lasts a lifetime. So it's produced in the embryo and you get it the whole time. Now, we have to be careful with this because there are homeostatic imbalances. For instance, glaucoma. And so basically, you block the drainage of humor and it increases pressure, and it can cause compression of the retina. Let me go block humor. Increases pressure, right? And it can cause compression of the retina and the optic nerve, and so that's what ends up leading to blindness. So let's go look a little bit at the next so that we can see some of this, and then we'll get on to talking about essentially the retina, the innermost layer. So this kind of shows you how you can differentiate the anterior segment from the posterior. And so essentially all you do is locate the lens. Anything in front of the lens is considered the anterior chamber, and anything that's behind the lens is considered the posterior chamber. So the aqueous humor is up here in this basically interior chamber in here. And then once you get behind the lens in the posterior part, you're looking at the vitreous humor. That's how you determine the front from the back. So let's look at some of these areas really quick. So we can, of course, the very outside you don't see here is the conjunctiva. And then if you look, this part right up here, that you see the arrow, that is the basically the cornea. This part that's white and not transparent but opaque is the sclera. You can see the choroid is right in here, and that was the one that essentially, when we were looking at it, that it supplied all the blood to the eyeball, and it had that kind of brown pigment to prevent light scattering and visual confusion. Ciliary bodies. You'll see that here. And those are essentially those smooth muscle bundles and ligaments that hold the lens in place and help to control the lens shape. Of course, this is the lens here. The iris, you'll see, is the colored point here. And basically, within the iris, here is the hole that's known as the pupil. And remember, it regulates the amount of light that comes in through muscle contractions. The lens is that biconvex, which means it goes out on both sides, right? And it can change light to help as light comes through to focus on the back in the area called the retina. And so, as you can see up here, anterior segment there that contains the aqueous humor, think anterior for A, and then towards the end of the alphabet is V, or towards the back is the posterior humor, and these basically help to keep up here. The vitreous humor is basically there to help maintain the shape of the eye. In the front, it's mostly water in the aqueous humor, but it also helps to essentially, it has enzymes and electrolytes and glucose that help to nourish the eye in the areas that are avascular, and it helps to maintain eye pressure. So if it can't drain it right, then it can lead to glaucoma, and this essentially leads to blindness because of the increased pressure in the eye causes compression of the retina and the optic nerve. So now let's look at the retina here, right? And the retina has two layers. It essentially has what's known as an outer pigment layer. And this, let me change this to black, prevents scattering. And then we have an inner neural layer. And basically this interneural layer is going to be made up of, and I'm going to go on to a different, three main nerve types. You have photoreceptors. And there are two types of photoreceptors. We have what are known as rods. This is used for dim light and peripheral vision out towards the side, right? AKA no color. And this requires vitamin A for it to work correctly. You also have cones, and think C for color. And so there are homeostatic imbalances for each. For instance, rods, you get, if they don't work right, you get what's known as night blindness. And this is for a lack of vitamin A. So if you take in more vitamin A, it should help your night blindness. And for cones, a disorder noted for them is color blindness. And there's nothing you can do about color blindness. It is basically you're missing certain cones, and this is genetic. It's actually X-linked recessive, which means if you're a man, you get it from your mother because men are XY, and you get it from here. If you're female, you'd have to get it from both your parents. So females are less likely to be colorblind. You also have what's known as bipolar nerve cells. And then ganglion. So basically, the information for light comes in through the photoreceptors, then goes to the bipolar cells, and they basically receive information. Info from the photoreceptors, and then they talk to the ganglion and they relay it to the brain. These guys exit the eye as the optic nerve. And basically, there's what's known as an optic disc. And this is where the optic nerve forms. There are no photoreceptors there. So let's go back to our picture. And so you can see here optic nerve. We also know that as the blind spot. So this is, as you can see, where all those ganglion cells are starting to come together. So this is where basically you won't see anything here. Now we do have a blind spot. Your brain's really cool because it integrates information that surrounds it and helps to fill it in so that you're not seeing a big black spot over in the corner. Now you have the Macula Lutea and the fovea centralis, and these are areas that that are high in photoreceptors here. And so we'll talk a little bit more about this when we get into the lab. And so this is just kind of giving you a photograph of the human eye so that you can actually see what it looks like, and we'll be dissecting a sheep eye in class. Now, we did talk quickly about cataracts, and cataracts happen whenever you have a problem with your lens. It's a homeostasis imbalance, and basically you get this clouding. So this is clouding of your lens, and it's basically with proteins that have been crystallized. This normally happens due to aging, diabetes, it can be more common in people that smoke heavily or exposed to intense sunlight.
This is Dr. Sibley. We're doing a cataract surgery today. This patient is legally blind, and we've given them some medication. They don't feel this, they don't see this. They're laying down comfortably, and I'm sitting at the head of their bed. This is the patient's left eye, and at that position I'm touching the eye with a diamond blade. That diamond blade is adjusting the astigmatism. This patient has a very gray cataract. You can see it inside the circle of the pupil. I've just entered with my left hand, and I'm going to fill it up with a capsule stain. That capsule stain will color the layer of the cataract. The cataract is kind of like a lima bean. The lima bean skin is very tough and hard to visualize. This is a clear jelly. This clear jelly is now filling that chamber of the eye, and it's pushing the blue food coloring dye out. We don't need that anymore. So we have colored the skin of the cataract, kind of like the lima bean skin, and the right hand is going to enter with a 2.5 millimeter entry blade, and then my right hand is going to use an instrument to grasp the cataract capsule, and that cataract capsule is being opened in a circle, kind of like a manhole cover, and we're going to get the lima bean skin, if you will, removed and an opening made, and then the gray cataract can be popped out of that cataract shell. I'm going to do that with a little bit of water in my right hand, and you can see it pop up, and it's very gray and it's very cloudy, and this patient was legally blind. This instrument is it's a tiny straw with a big name, phacoemulsification. It has water flowing in, it has a vacuum flowing out, and it has an ultrasonic tip on it, and that vibration will emulsify or liquefy that cataract. You can see that the cataract is being liquefied and vacuumed out with that instrument in my right hand. And so the cataract now is 90% gone all through that one straw. And that straw is finished, and I'm just going to show you that inside that remaining capsule bag, it's kind of like a Ziploc, and it's got a little bit of fibrous cloudiness, and that's kind of like a little wisp of cotton candy all around inside that. So it's kind of like a Ziploc that's still a little dirty inside. This straw in my right hand is removing, again, with vacuum and with the salt water flowing.
So that kind of gives you a little idea of how a cataract surgery works. So we're going to pause it here, and I'm going to give some time for you to kind of review over what we've gone. And when we come back to this lecture, we're going to hit on how light works and how we're able to see and how our eye accommodates light.